You are welcome. In today's class, we are going to look at the concept of virology. What is virology? Virology is a branch of microbiology that deals with the study of virus. And what is a virus? A virus is an infectious agent that have the ability to infect host cells, and the host cells can be plants, can be animal, can be bacteria. In fact, virus can infect other microorganisms. Like for example, there are some viruses that can infect fungi. There are some viruses that can infect bacteria like bacteriophages. And of course, this virus is made up of two, or in some cases, three major components. And these components include nucleic acid, protein, and envelope. One, the virus is made up of nucleic acids, protein coats, which is also called capsid protein, and then envelope in some virus. So it's not every virus that contains envelope. And there is a debate because it has been established that virus can only survive or can only replicate when found in a living host. But outside a living host, virus do not possess any characteristics of life. So that is why there is a lot of debates whether a virus is a living or dead. But as a result of that debate, scientists have reached an agreement. The virus can be in a borderline between living and not living, or chemistry and life. So as a result of that, virus is considered as in between living thing and not living thing. It can only be considered as a living when found on a living host. And outside a living host, it's completely not living. So actually virus exists as a borderline between living thing and non-living thing. And one very interesting thing that we should understand is that virus is very, very small. It is very, very small in the sense that the virus cannot actually be seen with a light microscope except when electron microscope is used, because it is very small. In fact, the size of virus is between 20 to 200 nanometer in size. Nano is very small. It's for -9. So when you said -9 meter, the size is very, very small. But compared to actually bacterial cells are usually measured in micrometer. Like for example, the normal range of bacterial cell is between 2 to 3 micrometer. So micro is power -6 and nano is power -9. So that means that approximately bacterial cells is 1,000 times the viral size. It means that bacteria is 1,000 times the virus in size. So that is why virus is very, very small. You cannot even observe it with a simple light microscope, except when you use an electrical microscope. So virus is very, very small. It's very, very tiny, and it's not actually easy to observe it with a normal microscope, unless when you apply electron microscope. That is the only way that you can actually see virus. And one very interesting thing is that the virus also have a gene, which also have the capability or the ability to mutate and evolve. Like for example, from the history, we know that coronavirus is a virus that actually normally found in bats. But because of the mutation they evolve, that is why they now jumped to be human parasites. So we should understand that it is very, very interesting to understand that viruses can undergo mutation and they can evolve. They can even jump from one particular host to another host. Like for example, in previous years, it has been known that the bat is the major host of coronavirus. But through mutation with environmental changes, the virus undergo some structural or some genome rearrangement, and it's now actually become human parasite. So it is possible. Virus can undergo mutation to actually jump from one particular host to another host, and that is what they call evolution. And of course, when you said evolution, is a process that involves the change of a living organisms over time due to environmental changes. So there must be a selective fragile. So it's a fragile that will make some of the virus to undergo mutation and evolve. And as a result of that, they can actually jump from one particular host to another. And of course, that is the virus. They have genes and they undergo mutation, and at the same time they can undergo evolution to produce another form that can actually jump from one host to another host, like in the situation that happened in the case of coronavirus. And then in addition, viruses have different structure. It ranges, it have different shapes. We have some viruses that have helical shape. Some of the viruses have icosahedral shape, and some of the virus structure is very complex. So viruses have different shape. It's not the structure of one virus vary from one another. So we have the major shapes of the structure of the major shape of virus is it's actually helical. We have helical virus, and we have icosahedral, and then complex structure. So these are the three major shapes of the virus. So we have, as I said, we have helical, we have icosahedral, and then finally we have complex structure. So these are actually the shape of the virus. And then of course now let's look at let's actually look at the structure of the virus very well. So virus is made of, as I said previously, is made of three major structure. There is viral genome, and then there is also a capsid, and then finally envelope. So let's start with actually viral genome. So virus generally contain only DNA or RNA. So in virus cannot actually contain both DNA and RNA like what happens in normal eukaryotic cells or in normal bacteria or in normal cells. So actually virus, they either have DNA or RNA. So the virus that have DNA is called DNA virus. And then the virus that have RNA, they are called RNA viruses. But one very interesting thing is that in virus, they can have they can have single-stranded DNA or double-stranded DNA. So the virus that have single-stranded DNA is called single-stranded DNA virus. And then also the virus that have double-stranded DNA is called double-stranded DNA virus, and also apply to the RNA. We can have single-stranded RNA in virus, and we can also have double-stranded RNA. So the virus that have single-stranded RNA, they are called single-stranded RNA virus. And then we also have double-stranded RNA virus. So virus can also be classified based on their genome. They can be single-stranded RNA virus, double-stranded RNA virus, single-stranded DNA virus, and double-stranded DNA virus. And their RNA or DNA can be in the form of either positive or negative. So we can have positive single-stranded DNA virus or negative single-stranded RNA virus. So virus actually, they can also be classified on the basis of their DNA. So that is actually for the viral genome. It's either contain DNA or RNA, but they cannot have both the two. You cannot have virus who have both the DNA and RNA. They only have one. So, and then the second structure of virus is a capsid. A capsid is actually a protein that usually cover the uh, the genome of the virus. It's like actually a pot that actually that assemble to cover the DNA or RNA of the virus, and that would actually help the viral, it help actually to protect the genome of the virus. So that is the major functions of the capsid. It protect the viral genome. So a capsid is a protein. So when you have the viral DNA or the viral genome together with the capsid protein is called nucleocapsid. Nucleo means nucleic acid, capsid means protein. So nucleocapsid, it means when you have a combination of the viral genome and the protein at the same time, so we have nucleocapsid. And that is why a virus is actually considered as not complete living organisms. The reason is because the virus is under or is a particle that fall under supramolecular complexes. And supramolecular complexes is just a combination of some macromolecules, like also apply here. Nucleic acid is a macromolecule. Protein is also macromolecules. So when you have macromolecules and another macromolecules joined together using non-covalent interaction, so the combination is called supramolecular complexes. And in addition, this nucleocapsid, especially the capsid protein, contain a protein that usually sometimes protrude or extended externally on the surface of the virus, which actually used for the attachment of the host cells, especially actually for the viral infection. For the virus to infect a host cells, there must be a protein that is used or that will be used in cellular recognition. So that proteins is usually like a ligand that used in recognition of the host cell's receptor, bind to it, attach to it, and then initiate the process of viral infection. So the nucleoprotein or the capsid protein, which is actually a coat that cover the viral genome, is also used in the attachment of the host cells for the viral infection. An infection. And then the next thing that we are going to look at is actually the spread of virus. So generally, a virus can actually spread from one organism to another organism, but it depends on the organisms. Like for example, virus can actually spread from plant to plant by vectors. That is, when we said vectors, they are microorganisms that transfer or organisms that transfer disease agent from one point to another. So virus can actually spread from one plant to another plant using the vectors. And in addition, especially in animals, virus can be spread through body fluids. Like for example, in the case of HIV, HIV can be spread through body fluids like semen, like sperm, blood, especially during sex. So HIV virus can be actually spread through body fluids. And then dengue. Dengue virus is a virus that actually can be spread from one person to another, but using a mosquito vector. Aedes mosquito and Felix, they are the species of the mosquitoes that transfer a dengue virus from one organism to another. And another example is actually rotavirus. A rotavirus is a virus that actually infects children through direct contact. So this is how actually some viruses actually infects one or infects infect one host from another host, either through direct contact, through body fluids, or exposure to the body fluids, or using a blood-sucking insect, like in the case of dengue. So then, actually, antibiotics doesn't work on virus. It doesn't work on virus. But there are antiviral drugs which are used specifically for the treatment of virus. So you cannot use antibiotics to treat viral agent or viral disease, but there are specific antiviral drugs that are used in the treatment of viral infection. And then in addition, also vaccines. There are a lot of vaccines that are available in the market for the prevention of the spread of the virus. And one very interesting thing that I have not mentioned is it's not actually in all cases that the virus are dangerous, because in some cases you can use virus in gene therapy. You can also use virus in vaccinology, that is in development of vaccine. So you should understand that there are some cases that you can use virus to actually help in curing some other diseases like gene therapy and development of vaccine. And then the next thing that we are going to look at is the viral infection. Viral infection. Virus actually infect hosts in different ways. So there are steps actually that involve in the viral infection. Number one, attachment. So first, the virus, there must be a cellular recognition. So there must be a ligand from the virus and then the receptor from the host cell. So the virus actually would use the receptor and attach to the surface of the host cells. Like for example, in the case of coronavirus, the ligand of the virus is spike protein, while the receptor from the host cell is angiotensin converting enzymes receptor 2, which the virus recognize, bind to, and then of course inject the genetic material into the genome of the host cells. So this is actually what happens. So in the case of in some cases, like for example bacteriophages, they usually inject their naked DNA into the host cells. But in the case of plants, virus, the virus that actually infect plants usually get into the plant through a process called endocytosis. So, and then after that is entry. So after the attachment of the virus on the surface of the host cells, the next thing is the entry. So the entry can either be using endocytosis, like in the case of plant virus, or the release of the naked DNA using bacteriophage. And then the next thing is replication and assembly. And then after that is aggregation and the boosting of the cells, where each of the virus will actually boost the cell. The cells will be degraded, and then the nearby cells will be infected with the virus, and then the virus will be continue replicating and affecting other cells until when the virus is actually overwhelmed the host cells. So ladies and gentlemen, this is actually a brief introduction of virology, and in the next video we are going to continue with the discussion. And thank you very much. And if this is the first time you are coming, please try to subscribe and join my YouTube community. Thank you.